Hello, everyone, and welcome back. I hope you had time to have a bit of lunch. Thank you so much for joining us today. It's great to see, about I think at the moment we've got 120 people on YouTube watching this, so that's great. I'm Linda Samuelsson. I'm a senior scientist at AgResearch, Palmerston North, although, of course, at the moment I'm working from home. And my partner in crime is Craig Pritchard from Massey University. So welcome back to this afternoon, the afternoon of the Sheep Milk New Zealand conference. And we're going to kick off here with session 4 today, which is the nutrition session. So we have several interesting presentations lined up for you. And the first one is Dr. Amber Milan from the Liggins Institute at Auckland University. And as you can see, Amber is not wearing her trek pants or onesie today. It's nice to, it's nice to have an excuse these days to dress up a bit and fix your hair. Yeah, isn't it? So Amber Milan is our keynote speaker. And since this is an online version of the conference, it's just a slightly shorter version than we had planned. So Amber will start to enlighten us all about all these things that we often talk about and hear about digestion of milk, about maybe milk intolerance, A2 milk, and all these things to make it a bit clearer for us all what this actually means. And then she will also present the findings from the world's first human clinical trial. So Amber, please.
Thank you, Linda. So as Linda introduced me, I am a research fellow at the Liggins Institute, University of Auckland, and I'm also a researcher at AgResearch as well. So I'm going to take you through, as Linda said, kind of a journey around milk digestion. Oops, sorry about that. So this is a little bit shorter than I was originally going to do. So some of this I may have to kind of snap through. But what I'd like to start is kind of with the background of our relationship with milk and how we digest milk and what that means in terms of digestion intolerances, but also allergy and how nutrients are delivered, and what kind of considerations we have across different types of milk. Then I'm going to take you through a couple of examples of that, which should give us some insight into what types of differences we might expect between sheep milk and cow milk.
This meeting is being recorded.
And so I'm going to take a few examples from some of my previous research, which has been done on goat milk and A2 milk, before bringing you on to the main event, talking about the sheep milk study that we recently completed. And finally, I'll finish off with a few considerations for kind of where to next with milk research. So to start off, you're hopefully aware that milk has a lot of different nutrients that are important for us. So things like essential proteins, fatty acids, and also many minerals that are important for our health. And there's good evidence that actually having a dairy intake in the diet can be protective for our health in a number of ways. So we see that right through chronic disease reduction, like type 2 diabetes, cardiovascular disease, and also the importance of dairy consumption on things like bone health. But despite these known benefits, there is a variability in how milk is consumed across the world. And this map on the side of the slide here just shows what the typical consumption, with dark blue being regions that typically consume a lot of milk, and other regions which don't traditionally consume as much milk. So despite these health benefits that we're aware of with dairy intake, then what might be explaining some of these differences in consumption patterns, and what does that mean for health? Well, there could be a number of reasons for dairy avoidance, but I'll kind of just talk about what we need to consider when we're talking about not having dairy in the diet. So obviously there are key essential nutrients that we need to get, but we could get them from other foods in our diet. But when we talk about dairy avoidance, we're also considering not only these nutrients that we might be missing out of, which could cause deficiencies, but there are also social impacts and quality of life impacts that avoiding foods can have. We can obviously get some of these nutrients from other components in our diet, but this can be quite challenging, especially in a modern Western diet. So if we take the example of a glass of milk and the example of calcium, which is an essential nutrient, the amount of calcium that you get from a glass of milk, to get it from vegetarian sources, you would have to consume a huge amount of these. So this is just an example of spinach or beans. And so for some people who may not be able to get these nutrients from other sources in their diets, it can actually be a risk factor for them having insufficiencies in their diet. But we know actually that regardless of this importance for health, dairy avoidance is an increasing trend. There's a recent report that shows that 1 in 6 Australian women actually actively avoid dairy consumption. So what we need to do is really understand why people are avoiding dairy so that we can understand, well, how can we actually make this maybe a workable part of their diets, or find alternatives that are actually going to suit them without putting them at risk of ill health. So the first thing to understand when we talk about avoidance is why people avoid it. Well, in that study that I just cited, talking about 1 in 6 Australian women avoiding dairy, 80% of those people who avoided it for biological reasons. So talking about negative reactions that they had when they consumed dairy. So when we talk about negative reactions, we can kind of put them into 2 different camps, so either an allergy or an intolerance. But it's important to understand that these have very different reasons behind them, and so therefore very different diagnoses and very different ways that we treat them. So for something like a milk allergy, or even allergy in general, we know that this is an immune response from the body, and it's usually as a result of a protein that's in a food. And so that's why we see that milk is actually quite a common allergen, as well as some of these other ones that you may be familiar with. This is more common in children, and as the immune system develops, this can actually be lost later on in life. So we see that for adults, food allergy is actually far less prevalent. Intolerance, on the other hand, is not really caused by a protein per se, but it can be caused by a number of different components in food, such as the carbohydrate component or different chemicals that are in the food. And we find that actually the prevalence of intolerance is much greater in the population and may actually increase as we get older. But importantly for intolerance, because there isn't an immune function related to it, it can sometimes be a little bit more difficult to actually pin down what the trigger is as part of an intolerance, because there might not be a really clear mechanism on how that happens. So for milk, what we know, luckily, is that lactose intolerance is the most common type of milk intolerance, and we actually understand this quite well. So there's a lot that we can do in terms of research and understanding this and helping people with that. So lactose is the main sugar that's in milk, and it's composed of 2 different monosaccharides, glucose and galactose. And when it's normally digested, the enzyme that's in the small intestine, lactase, will break down that sugar into its component parts, which can then be absorbed. But when there's not a sufficient amount of that lactase enzyme, then what happens is that lactose isn't broken down, and it transits all the way down to the large bowel, where it's fermented by bacteria to produce a range of gases, short-chain fatty acids, and these are actually what's responsible for a lot of the discomfort that we associate with something like lactose intolerance. So it's that inability to digest lactose. We also see that this has quite a large prevalence across the world. It's estimated about 75% of the world's population is lactose intolerant or a malabsorber of lactose. And this is a bit of an inverse map to what we saw with the consumption patterns of dairy, showing that actually in a lot of those places where dairy consumption isn't habitual, that's where we actually see lactose intolerance. And part of this actually comes down to the genetic basis of that loss of ability to digest lactose that we see in actually a minority of the world's population. But what we also know is that it's maybe not quite a static situation, because we do see that some of these regions that we know are largely lactose intolerant will still consume dairy. And that comes down to how that lactose is fermented in the gut by the gut bacteria and what types of responses that induces. So this is where we're kind of starting to understand that actually the gut microbiome plays a really important role in that link between the malabsorption of lactose and actually causing an intolerance. But coming back to some other reasons why people might avoid milk, we actually know that lactose isn't the problem for everybody. So here is some data looking at people who self-report that they are lactose intolerant, and looking at how much lactose they ferment when they're given it. So just an indicator that they're malabsorbing lactose. What we see is that there's a subset of people that even though they think they're lactose intolerant, don't have this typical rise in breath hydrogen that we would expect that indicates that lactose is not being digested and is instead being fermented. So when they're referred for a lactose challenge, they actually don't see any kind of a rise in malabsorption and don't have the associated pain as a result of lactose. But importantly, clinically, what this means is that if these people, represented by the black bars here, then go on to remove lactose or dairy from their diet, we actually see that it doesn't do them any benefit. So if someone with lactose intolerance were to remove lactose from their diet, we can see that that can improve their symptoms or their tolerance of foods, but we don't see any improvement for someone who's not got lactose at the heart of their intolerance problem. So I think this really highlights that we need to understand why someone's avoiding a food and what the mechanisms are so that we can understand what specific foods might be suitable alternatives for them. So this is where it gets a bit more interesting, because if it's not lactose that's the problem, then what could it be? Well, we know that there are lots of different components of milk, so I've kind of touched on the carbohydrate component, but there's also a large amount of proteins and fats, which influence how milks are digested. So focusing on protein, we know there are two main protein components, the casein and the whey fraction of milk. And within these, there are multiple different subsets of these proteins, which have different physiochemical properties. When we look at allergy, we know that allergies to milk are usually caused by these specific proteins that are found in milk. And this is through that immune response, an IgE-mediated immune response. But what we also know is that proteins have a lot of different biochemical functions, and we actually see that there are bioactive properties of a lot of these other proteins, particularly within the caseins. But these can exert effects on the immune system as well, which could be non-IgE mediated. There's also a huge amount of impact that proteins have on our digestion, which I'll talk about a little bit more in the next couple slides. So proteins can have a really important impact on the digestion of milk and then how we perceive that digestion. Now, it gets even more interesting when we start to think about what the variation in those proteins are. So not only do we have these different types of proteins, but the proportions of these different types of proteins vary widely across species. So this is an example just of casein. And you can see that if we look across from bovine through to ovine, you have this change in the amount of something like beta casein that's found in different species milk. Along the bottom here, I'm just showing what the sequence, the protein sequence, the amino acid sequence of that beta casein looks like. And you'll see that across cow, sheep, and goat, you've got slight variations in the actual sequence of those proteins. And what that means is that they're going to have different ways that they interact with each other and also interact with the body as they're digested. Not only do we have these differences across species, but actually within individuals within a species, you can have variation as well. So you're probably familiar with A2 beta casein, which is thought to be the ancestral beta casein that cows had, and most other species have an A2-like beta casein as well. There's a variation in some herds where that protein sequence has changed, which results in a different cleavage of the peptides that are formed when this is digested. And those can have very different effects on both the immune system, but also on how that's digested. So to give an example of how this changes digestion, this is a really well-known example of fast and slow proteins. So if we look at the dynamics of how whey and casein are digested, what we see is that casein is thought of as a slow protein, whereas whey is thought of as a fast protein, meaning that the amino acids that are in that protein are released into the body much more quickly than they are with casein. We also see that this results in a much greater increase in those proteins within the muscle tissue as well, which is quite important because having those amino acids from a meal getting into tissue is what actually drives something like muscle growth. So the reasons for this, there are some physiochemical reasons behind this. So we know that when curds and whey are formed, we've got a solidification of the casein protein. And when that occurs in the stomach, that changes the dynamics of that protein digestion. But there may also be some biochemical reasons for this as well. So as we discussed with those bioactive peptides that are released from certain casein molecules, that can actually have an impact on gastrointestinal transit as well. But it gets a little bit more interesting as well when we think about the structures of these proteins. So here's an example of gels, or the curds that are formed, which is similar to what would happen in the stomach, but can also be manipulated through food processing techniques. And so although we know that casein should be digested a bit more slowly, what this is showing is that when we have a gel formed of whey, so these red and pink bars, we actually have a lower release of the amino acids that are released as a result of that gel structure. And we can see, looking at that gel structure, there's an interaction between the proteins and the fats in that gel structure, and that changes the way that they're accessible to be digested and therefore their response within the stomach. Fat structures can also be quite interesting because they're quite complex as well, having things like the milk fat globule membrane, which can interact with proteins. So all of these features interact to actually change the way that these milk structures are digested when they're eaten. So that's just a bit of a background, but how do all these ways that composition can impact digestion influence how different species milk is digested? So I'll take you through some of our clinical research looking at the differences between goat and cow milk, and also looking at that species differentiation, or sorry, that interspecies differentiation with A2 milk. So the first study was looking at a comparison of a growing up milk formula based on either goat milk with a whole protein cow milk. And we also used as a comparator a hydrolyzed cow milk, which is thought to be where the proteins are broken down and thought to be more tolerable to infants that don't have good tolerance to cow milk. What we did in this study, which is a similar design to the rest of the studies that I'll talk to you about, was to provide our participants with a single glass of milk and follow up after a period of several hours to look at both their biological responses, so in terms of nutrient responses of fats, proteins, etc., and also looking at their tolerance, so looking at that malabsorption of lactose, as well as the symptoms that they felt. So what we saw was that comparing goat milk, which is the blue lines here, with cow milk, the black lines there, there's actually a very similar response. This is looking at the rate of digestion of that milk between those two whole protein milks, even though there are quite different compositional differences between them. But with the hydrolyzed cow formula, which had a different protein composition and a slightly different overall composition, this actually delayed the emptying of the gastric contents quite substantially. And we saw this follow through as well in terms of malabsorption. So regardless of cow and goat milk, there was a similar amount of malabsorption, but this was much more reduced with the hydrolyzed cow formula. And similarly, we saw not really much of a difference in terms of tolerance between goat and cow, and not a difference with the hydrolyzed formula either. When we looked at what this impact of the digestive response had on the absorption of nutrients, what we found was that, again, we saw a much more different response with the hydrolyzed formula than we did between species. So this is the insulin response, kind of measuring that body's metabolic response to these milks. And what this also resulted in was that we saw this really, really rapid decrease in the amino acids, these branched chain amino acids, crucial for muscle growth, that appeared in circulation following ingestion. So this shows us that not only does the protein structure have an impact on the rate of digestion, but it also has an impact on the delivery of those nutrients. This second study, the Amigo trial, looked at the differences between A2 milk and conventional milk containing both A1 and A2 beta casein. So what we really wanted to get to at the bottom of this study was to understand how people who might be intolerant to milk, but not lactose intolerant, how differences in the protein structure, beta casein structure, might impact their tolerability of milk. Thinking that if it's not lactose, well, it must be something else. And so is it the beta casein portion of the milk? And so what we really wanted to understand was that difference between the lactose-mediated and protein-mediated intolerances, because we know that A1 beta casein has been linked to changes in digestive rate and also immune markers being elevated. So what we did was we chose to have a dairy tolerant group to act as our control. And we recruited a group of women who were self-reported intolerant to milk and determined whether they were either lactose intolerant or lactose tolerant. And then we provided them in a randomized order either conventional milk, A2 milk, or a lactose-free conventional milk. And again, we looked at a number of biological markers to see what the availability of nutrients was, as well as reported their symptoms and malabsorption. So what we found was that for people who had lactose intolerance, they had typical symptoms that we would expect. So they had an increase in cramping, things like flatulence after drinking. And this occurred quite a long time after, because it does take a little while for that lactose to reach the large bowel and to actually exert those symptoms. So it's really after about 90 minutes plus. What we also saw was that for the non-lactose dairy intolerant group, so they were tolerant to lactose, they had symptoms as well when they drank milk, but they were a different set of symptoms. So they had these upper GI symptoms that were which were characteristic of bloating, distension, belching. And we also saw that these symptoms actually resolved, whereas for someone with lactose intolerance, those symptoms just continue and actually get progressively more intense the closer we get to sort of 3 hours. So this really suggests that there is some kind of a different mechanism at play causing this intolerance. And so the question is, well, if we change the protein in the milk, is that going to change the tolerability of that milk? What we saw was that for people who didn't have lactose intolerance at the heart of their intolerance, changing the beta casein structure in the milk didn't actually provide them any relief. So they didn't have an improvement in symptoms like flatulence. But what we did see, surprisingly, was that for people who are lactose intolerant, changing to A2 milk, changing the protein actually improved their tolerability of milk, regardless of the fact that there was actually lactose in that milk. So we can improve fecal urgency and nausea by changing the protein in the milk. And this is accompanied as well by a reduction in that amount of malabsorption that's occurring. So again, we're looking at probably different mechanisms, but it is also showing us that changing the protein in the structure of the milk does have an impact even for people who have lactose intolerance at the basis of their intolerance. So just to sum up very briefly, clearly protein has a really important role to play here. It could be a mechanical or an immune role. And there are lots of different things to think about in terms of interactions between these components of milk that have a really important feature. So to bring you on to the sheep milk digestion study that we've gone on to do, I'll be talking mostly about the nutrients and tolerance that differences that we saw here. So the design of the study was very similar to the other studies that we had, where we have a milk challenge and we follow up with looking at nutrients and tolerance over the course of several hours. For this study, we decided to do a glass-for-glass comparison to understand what the impacts are, comparing completely on a usual serve basis. So you will note that actually sheep milk is much richer in protein, fat than cow milk is. And we actually also had a bit of a difference in the lactose content. So not only do we have these compositional differences in the types of proteins, the types of fats, but there's also a proportionality difference in terms of how much is actually available. For this study, what we decided to use was a subject group that was self-described as a dairy avoider. So these women said that they avoided dairy, and when we actually looked at their characteristics, we found that a number of them had underlying lactose or food intolerance issues characterized by irritable bowel syndrome, so sort of a generalized digestive discomfort. And about 80% of them were lactose intolerant as well, which is not surprising, given that that's one of the main reasons we see people avoiding dairy for. But when we looked at whether the nutrients in sheep milk are digested differently or absorbed differently from cow milk, what we found was hopefully not surprising. So this is looking at the amino acids, particularly those essential amino acids and branched chain amino acids that we see in sheep milk, and looking at a comparison between the sheep milk over the course of those 5 hours and cow milk over the course of those 4 hours. So sheep milk has more of all of these amino acids compositionally in the milk than cow milk does. So in a single glass serve, sheep milk was providing more of these amino acids. And what we saw was that indeed these were showing up more abundantly in circulation, so in the blood. They were getting absorbed well, and they were showing up at higher concentrations than for cow milk. Importantly, we see that as well for these branched chain amino acids, and recall that these are really important for things like muscle growth and muscle maintenance. So on a glass for glass basis, you can get more of these essential and branched chain amino acids in circulation than you would for a glass of cow milk. Coming on to fats here. So I haven't talked too much about fats, but they are quite complex and they can have interactions as well in different ways that they're absorbed. So when we look at the sheep milk response, the first notable thing is that this overall blood triglyceride response, so the amount of fats that are in blood, looks pretty identical to between the sheep and the cow milk. Now, this is actually surprising because if you recall, the sheep milk has more fat per glass than cow milk does, and yet we're seeing the same fat response in the blood. Now, this is quite an important thing because we know that high-fat foods can trigger a really exaggerated response of this, this blood triglyceride response. And we also know that having an elevated triglyceride response for a long period of time is a potent risk factor for cardiovascular disease. So even with more fat, the sheep milk is delivering the same response in the blood for triglycerides. Part of the reason for this is probably because sheep milk is more abundant in medium chain fatty acids. So we see that these medium chain fatty acids also appear in circulation at a much greater proportion following sheep milk than they do with cow milk. And these medium chain fatty acids are actually digested differently from long chain fatty acids. They're digested really rapidly, and they don't go through the same complex metabolic processes that long chain fatty acids do. So this is probably one of the reasons why we're seeing the same triglyceride response is because those medium chain fatty acids are really bioavailable from that sheep milk, and they're not causing the same elevated spike in triglycerides. So was sheep milk better tolerated than cow milk? So what we looked at was that malabsorption, and recall most of these women were lactose intolerant, and also how they were able to tolerate it based on their symptoms. So we found that for sheep milk, there was less malabsorption than there was for cow milk. But when we did the analysis of the sheep milk, we also found that actually that sheep milk had less lactose than cow milk. So this is a little bit unexpected because you would usually expect that that lactose content would be quite similar. And some of the reasons might be because we used a powdered sheep milk that was reconstituted. But it does raise the question is whether this reduction in malabsorption is just because of a slightly lower content, or are there other specific differences in the way that milk is digested that contribute to this? What we didn't see was any change in digestive comfort. So after both sheep and cow milk, women reported the same amount of discomfort. Not surprising because they were lactose intolerant, but it does raise the question about whether people who are tolerant to lactose but maybe intolerant to milk might have a similar response or a different response to sheep milk versus cow milk. So this is just a summarizing slide to bring up the point that we saw that the proteins were more readily digested. There's a different fat response with sheep milk, and that there also could be a reduction in lactose malabsorption, but definitely something that we need to look at in more detail. So this has all brought a few questions with respect to sheep milk. So what about different tolerance states, obviously? Or are there differences with longer term consumption? So might tolerance be improved? What impact does that have for nutritional outcomes of these key nutrients? And what about different types of sheep milk formats? So to kind of give a picture of where that might go, I'm just going to briefly touch on some of the other work that I've been doing looking at sort of these long-term impacts and the importance of food structure. So one of the things we know is that the microbiome is really important to how we tolerate lactose. And we also know that repeated exposure to something like a prebiotic, so this is looking at results from a study that looked at a prebiotic in people who are lactose intolerant. After a period of time, this can actually improve their tolerance to lactose. And this is concurrent with changes in the microbiome. So there is actually a period of adaptation or an ability to adapt that tolerance to lactose, even if someone is malabsorbing, which is, I think, really important when we consider that 75% of the world's population is intolerant. So one of the studies that we've been doing to look at this is really to look at long-term exposure of milk in people who have intolerance to see whether changes in the protein structure can change how that milk is digested, have any impacts on the immune system, but also look at changing the microbiome to be able to better tolerate that milk. Another project that I'm involved in is the New Zealand Milks Mean More, which is hosted by the Riddett Institute. And this is really a multifaceted project to look at how processing of milk changes the way that we digest it and how this difference changes across species, and really targeting how we can get the most out of milk in terms of delivering nutrients for added health benefits. And it's a similar aim or similar approach to how we've looked at clinical research in the past, looking at a multi-omics structural approach, but really looking at how the processing techniques can actually impact health outcomes. Because we know that things like the species differences or even changes to the food structure can have those impacts on the digestive mechanisms and also through to those biological markers and functional health impacts. So the first study that we're doing as part of this work is to look at a simple processing technique, looking at pasteurized milk and UHT milk, and to see how that impacts digestion, which we're doing through a process of MRI imaging of the stomach, and to also look at how that impacts the delivery of nutrients. So really what we aim to do here is to get a complete picture of how that digestive process is impacted by food processing, validated across in vitro and preclinical models, and hopefully this can both inform future product development, but also applications that we can use to look at species-specific differences in milk digestion as well. So just to summarize, I think that we have a lot of insights into how milk is digested, that there are important implications for processing and species-specific differences that we should probably be exploring a lot more. And there's also a lot more that we can learn about dairy intolerances. So I don't think we've nailed down all of the mechanisms of dairy intolerance, and there's probably a lot more that we can do with milk or milk products to look at ways that we can maybe provide solutions for people and ultimately use it to improve overall health. So I'd just like to acknowledge the huge amount of work that's gone into all of the research that I've talked about today, both from my participants, my colleagues, my funders, and I'd just like to say thank you for listening.
Thank you so much, Amber, for this very interesting presentation. I just find it absolutely fascinating that you change something in one part of the milk, which is not lactose, and that affects how the lactose is digested. It's it's absolutely fascinating, and also just how the structure of the food also has an effect on, for instance, how the lactose and other parts of the milk are digested. It's and clearly this is a very, very complex area. So thank you for enlightening us and helping us to learn a bit more about this very complex subject.
My pleasure.
Yeah, so we have a couple of questions that have come in here. The first one is from Alistair Ross from AgResearch, who's asking if you measured glucose and insulin in response to the different milks, because usually higher branch chain amino acid response would lead to increased insulin secretion.
Yep, so we did. Off the top of my head, what I recall is that we didn't have a difference in insulin secretion between sheep and cow milk. We did have slight differences in glucose levels, but that actually resulted in a more stable glucose response with the sheep milk than we saw for the cow milk. So there was a bit more fluctuation with cow milk, whereas glucose remained a bit more stable. So I think it's a challenging thing because, of course, branched chain amino acids will impact glucose secretion, insulin secretion and glucose metabolism. But we also have the complexity where we've got different macronutrient and energy components of both sheep and cow milk. And what we've seen previously, so in the goat milk study that we looked at, that hydrolyzed cow formula that we used had a higher energy and a higher carbohydrate content than the others, which also impacted how that metabolic response. But I think what that highlights is really that we shouldn't be looking at the individual compositional differences to get a good picture of how that's going to act in a living system. We need to understand how those overall differences and how people would actually eat those foods, how that's impacting them, because that's really, at the end of the day, how it's going to actually impact their health.
Thank you, Amber. We also have another question that's come in here, and it's from Jessica Gathercole, also at AgResearch, and she's asking, did the amino acids in the ShinDig trial include all amino acids or just free amino acids?
So that's free amino acids. So in circulation, we were just looking at free amino acids. So of course, that won't take into consideration peptides or bioactive peptides that could be absorbed. But with the composition of the milks, we did look at total amino acids. So that's after the milks have been hydrolyzed to look at the full suite of amino acids.
Okay, thank you. So to everyone out there on YouTube, if you have a question, please post it on the chat and I will read it to the presenter. So yeah, I would like to thank you very much, Amber, for your enlightening presentation. And we have a few more presentations in this session, and they all kind of follow up on this theme. So it's good to have had this introduction from you. So let me introduce to you our next speaker, and that is Natalie Ahlborn. She is a student at Massey University and AgResearch, and she's presenting some of the work she did about a year ago, also looking at digestibility of sheep milk, but she's been using a rat model. So Natalie, over to you.
Cool, thank you, Linda. So I'll be presenting the results from a study investigating the in vivo digestion of sheep and cow milk in a rat model. So I'll just go over our hypotheses and the aim briefly. I'll just sort of touch on what the rat study itself come from, what it was made up of. I'll go over some results and then discuss a couple of interesting metabolites that we found. So anecdotally, consumers of sheep milk report less gut discomfort than those consuming cow's milk. And as a result, many of these consumers choose sheep milk as an alternative to cow dairy products. However, the reasons for these physiological effects are unclear and some hard evidence is needed. So the purpose of this study is to add to our fundamental knowledge and further our basic understanding of the digestion and assimilation of sheep milk in the human body. In a macro sense, sheep and cow milk are relatively similar. However, there are a number of key compositional differences which set them apart. A number of these Amber's already discussed. Previous work comparing goat milk and cow milk have shown that compositional differences between the milk types are enough to lead to differences in digestion. So our hypothesis was that the differences in sheep milk and cow milk would be sufficient to result in differences in digestion and assimilation in the body. Our aim was then to investigate these differences in a rat model using nuclear magnetic resonance spectroscopy. I don't know how to go next slide here. There we go, sorry. So this study used a rat model for the milk digestion and assimilation, and we used blood serum and fecal contents to gather our data. The rat is an accepted model for human digestion, and several studies comparing human and rat digestion have been carried out. So this study used 36 rats in 3 groups of 12. These rats were fed either sheep milk, diluted sheep milk, or cow's milk. The diluted sheep milk was diluted to have the same total solids as cow's milk. So over a period of 28 days, these rats had free access to the milks, and they were also provided with a dairy-free rat chow, although the rat chow was removed 2 days prior to sampling. After 28 days, we collected samples from the cecum, which is at the start of the large intestine, and blood serum. The fecal contents represents digesta after everything has been absorbed from the small intestine and before too much microbial modulation is carried out in the colon. So serum is blood without the clotting factors and is a transport medium around the body. This means the metabolite profile of the serum includes both end products and intermediates of metabolism in different organs and tissues. We then analyze these samples using nuclear magnetic resonance spectroscopy, which is a powerful tool often applied to biofluids and tissue for the detection and quantification of metabolites within a sample. So all of the NMR spectroscopy work was carried out at Massey University in Palmerston North. So coming to some results, looking at the fecal contents, we identified 37 metabolites. This included 18 amino acids, 6 sugars, 5 short-chain fatty acids, and 8 other compounds. We carried out a multivariate analysis which showed 8 metabolites with significantly different concentrations between groups of rats fed sheep milk, cow's milk, or diluted sheep milk. So these were propionate here in the dark red, phenylalanine here in bright red, lactose in yellow, 2-hydroxyisovalerate in green, dimethylsulfone in lighter blue, aspartate here in the medium blue, and 4-aminobutyrate in the darker blue. So here are the box plots to show the distribution of the data. All 8 of these metabolites showed significant differences in concentrations in a comparison between rats fed sheep milk and rats fed cow's milk. The sheep milk fed rats had high concentrations of aspartate, phenylalanine, propionate, dimethylsulfone, and succinate. Those fed cow's milk had higher concentrations of 2-hydroxybutyrate, 4-aminobutyrate, and lactose. It's also interesting to see that in general, the fecal metabolite concentrations of rats fed diluted milk sits in between those fed cow's milk and sheep's milk. So these 4 metabolites showed statistically significant differences in concentrations in a comparison between diluted sheep milk and cow milk. So dimethylsulfone and succinate concentrations were both significantly higher in diluted sheep milk fed rats, whereas 4-aminobutyrate and lactose concentrations were significantly higher in cow milk fed rats. When comparing the fecal profiles of rats fed with sheep milk and rats fed diluted sheep milk, only one metabolite showed significant differences in concentration, and this was propionate, which is found in higher concentrations in the sheep milk fed group. Coming now to the serum results. In the serum, we identified 29 metabolites and putatively identified another 2. So this included 14 amino acids, 3 sugars, 3 short-chain fatty acids, and 11 other compounds. A multivariate analysis then showed 10 metabolites with significantly different concentrations between groups of rats fed sheep milk, cow milk, or diluted sheep milk. So as we can see here, we have leucine in the dark red, isoleucine in the brighter red, 3-hydroxybutyrate in orange, fucose in yellow, dimethylsulfone is this thin green bar here, betaine is this lightest blue, and then we have acetone, acetoacetate, and acetate. So again, some box plots to show the distribution of the data. Here we saw significantly higher concentrations of isoleucine, leucine, and dimethylsulfone in rats fed sheep milk, and significantly higher concentrations of 3-hydroxybutyrate in the group of rats fed cow's milk. So this is in a comparison between the group of rats fed sheep milk and cow milk. This time comparing the serum profiles of rats fed diluted sheep milk with the rats fed cow's milk, we see significantly higher concentrations of dimethylsulfone in the diluted sheep milk group, and higher concentrations of 3-hydroxybutyrate in the cow milk group. Interestingly, comparing the sheep milk group with the diluted sheep milk group showed all 7 of these circled metabolites had significantly higher serum concentrations in the diluted sheep milk group than in the sheep milk group. So looking now at some interesting results, we see dimethylsulfone, otherwise known as methylsulfonylmethane, or MSM, was found in significantly higher concentrations in the sheep milk and the diluted sheep milk groups than in the cow milk group in both sample types. MSM is a naturally occurring sulfur compound commonly found in milks. It has been shown to have anti-inflammatory and antioxidant effects and can be found as a supplement in health food stores. It has been shown to be effective in the management of rheumatic and arthritic pain, in the reduction of knee cartilage degradation, particularly in osteoarthritis, and it has been shown to mediate autoimmune diseases. Milks are known to be a direct source, with the correlation between milk consumption and an increase in blood MSM levels having been reported. With this in mind, it is likely that sheep milk is responsible for the higher concentrations in serum and fecal material, and these results suggest sheep milk may have the potential to benefit individuals with arthritic pain and joint inflammation. So both leucine and isoleucine were found in significantly higher concentrations in the sheep milk group than the cow milk group. Both leucine and leucine are branched chain amino acids, otherwise known as BCAAs, and Amber has discussed these a little bit earlier. So BCAAs have diverse roles, such as in protein synthesis, the immune system, energy production, and neurotransmitter synthesis. These effects are accredited to all BCAAs, but are thought to be mainly due to leucine. Leucine also plays a role in the regulation of blood glucose levels. So these results reflect the findings of some HPLC-free amino acid work, which was done, and similar results were also found in Amber's human study. One difference here is that this rat study was a long-term study, while Amber's was acute. So the milk powder amino acid composition was analysed, and this revealed higher leucine and isoleucine concentrations in the sheep milk. This gives us an indication that the higher levels found in serum are likely linked to the higher levels in the sheep milk, and hints at the bioavailability of these essential amino acids. These results do show us that the sheep milk does increase circulating branch chain amino acids. So I've included this as it was an interesting result, and also because it sort of mirrors results seen in Amber's study. So lactose was found in significantly higher fecal concentrations in the cow milk group than in the sheep milk group. Lactose is a milk sugar and is broken down into glucose and galactose, both by the body itself and microbially. Because it is a sugar, it is usually broken down and absorbed fairly quickly. A macronutrient analysis of the milk showed sheep milk had a slightly higher lactose content than cow's milk, and that is the milks that these rats were specifically fed. At this stage, we don't know the reason behind the higher fecal lactose level in rats fed cow milk. So in conclusion, we see that the digestion of sheep milk and cow milk result in differing metabolite profiles, and in some cases this is a direct result of milk compositional differences. These differences raise the potential for sheep milk and cow milk to have differential effects on human health. Two good examples from this study include the potential of sheep milk to positively influence arthritic pain and inflammation due to higher MSM levels, as well as increasing circulating amino acids. It is, however, important to note that this was a rat study, and while a rat is a good model for humans, we can't directly extrapolate these results to humans without a human study. So on that, I'd like to wrap this up. A big thank you goes to Linda Samuelsson for her enthusiasm and support throughout this project. I'd also like to thank our key stakeholders, the Ministry of Business, Innovation and Employment, Maui Milk, Spring Sheep, and Kingsmead Cheese. Thank you.
Thank you very much, Natalie, for a very interesting presentation. I think this again, we saw lactose popping up there again with showing differences from the different milks. And I think that just adds to what Amber was saying before, that it's not just about how much lactose is in the milk, it's about what else is in the milk and sort of what format the milk is in. So adds to the confusion, which is part of doing research, that sometimes we end up with more questions than answers. That's just the way it is sometimes. I would just like to follow up a bit. I found this dimethyl sulfone very interesting. And first of all, we have a question that has come in from Andrew Galloway from Federated Farmers. He's wondering if you have any data on the naturally occurring MSM levels in sheep milk.
Yeah, I mean, I think that's a good question. I think it's important to remember that, you know, the presence of some endogenous or microbially produced MSM shouldn't be ignored. And there isn't really any existing data in the literature on the naturally occurring concentrations of MSM in sheep milk. But the milks from this study, so from this rat study, are undergoing NMR, so we can work out the existing levels of MSM in the sheep milk itself. I think this will also help us to elucidate how much of this effect is directly due to the milk itself and how much is a result of the body's processing of the milk. Okay, thank you. And I would also like to, I think it's quite interesting, the potential here of MSM that in sheep milk, that it might be something that is beneficial for people with joint problems. How would you take this research one step forward? Do you have any thoughts on that? Well, something I would really like to do would be, and I think it's probably the next step, is see if we can detect something in, for example, the serum of pigs. I mean, pigs is again one step closer to humans, and I think that would be a good place to start looking. From there, potentially seeing what we could find in a human clinical study would be awesome. But I mean, that's all sort of a question for the future. I think the first step, of course, is to find out what we know about the milks that we used in this particular rat study. So the NMR results from that will be really interesting, and then go from there. Okay, great. Thank you very much, Natalie. And we're going to move on now to our next presenter, who is PhD student Zheng Pan from the Riddett Institute. And Zheng, I know that you're going to talk to us about some in vitro experiments that you have done with sheep milk, and that will add to this body of evidence of how different milks are digested. So, please, Zheng.
Thanks, Linda, for your introduction. Yeah, today I'm going to introduce something about the impact of processing on the in vitro gastric digestion of sheep milk. And as we know, milk as an important source of protein for humans has been widely studied in both in vivo and in vitro digestion. And so far, plenty of digestion work on bovine milk has been widely investigated. But as the digestion of non-bovine milk, like human goat, sheep milk, and so on, is less studied. And it's widely known that the bovine milk and sheep milk are very different in their composition. As shown in the table, we can find the sheep milk has higher total solids than cow milk, and the content of fat and protein in sheep milk is nearly twice the value in cow milk, which may cause different digestion behavior within the gastrointestinal tract. And there are two main factors affecting the digestion behavior of milk, compositions and structures. As we know, milk is commonly exposed to the processing treatment, which can cause some changes in the structures of fat and protein in milk, and therefore resulting in the different digestion behavior within the stomach and intestine. So to be able to study the gastric digestion of milk, we use the in vitro gastric digestion model, human gastric simulator, to mimic the gastric digestion. And as shown in the interior view of the HGS, we found that it's composed of several units like the pumps, driving system, rollers, and the stomach bag. The HGS could simulate the biochemical processes as found in vivo. The food could be digested within the stomach bag. Besides, the HGS could also mimic the peristaltic movement of the stomach, and we can see in the lower image. The HGS could drive the rollers to squeeze the stomach bag and exerting the physical forces on the ingested food bolus and break them down into smaller particles. It has been proved that the HGS could simulate the physical forces as found in vivo. So, some digestion work on raw sheep skim milk and whole milk has been done by my colleague Debashri Roy before, and my work is follow up to Debashri's work, and the GI, as we can see, the GIF shows the milk how it digested within the HGS. Here is my protocol. Fish sheep milk was processed by several different processing treatments like the pasteurization, homogenization, and higher level of heat treatments. And these milk samples were digested using the HGS, and the resultant curd and digester was further analyzed by the following techniques. Here is the result. The figure shows the pH changes of the digester collected at different digestion time. As shown in the figure, we found the pH decreased gradually as the digestion time increased in all milk samples, but it shows differences in terms of the rate of decrease in pH. We found the thermoprocessed or homogenized milk could slower the decrease in pH, and the appearance of curd was also shown in the image. We found at 20 minutes of digestion, the raw milk formed a firm curd with a smooth surface, and with further digestion, the curd became smaller and its structure became more condensed. And when we look at the pasteurization, at 20 minutes, it also formed a firm curd, but with more porous and less integrated structure. And with further digestion, the curd was broken into several pieces at the end of digestion. About homogenization, we found at 20 minutes, the part of the curds was broken into crumbles, and with further digestion, the amount of the curd decreased significantly compared to raw milk and pasteurized milk. And for the higher level of heat treatment, we found at 20 minutes, the curd structure is much looser and more fragmented. It also has the crumble structure. And at the end of digestion, those crumbles became much smaller compared to pasteurized and homogenized milk samples. So we conclude that the processing treatments can make the curd's structure much looser, more fragmented, and less integrated. The curd's content was also analyzed. The figure shows the total solids of the curd. We found in the figure the total solids of curd decreased progressively as with increasing the digestion time, but it shows significant difference after 120 minutes of digestion. The total solids in homogenized milk samples decreased more rapidly than unhomogenized milk samples. And based on the total solids, the moisture content of the curd was also analyzed. We found in the figure, before 120 minutes of digestion time, the moisture content decreased in all milk samples, but after which the value remains roughly unchanged in all milk samples. It also shows differences in the moisture content, which means the processing treatments can result in different level of hydration of curd throughout the periods of gastric digestion, and which shows in the following order. So this curd's content result shows the processing treatments can change the curd structure and composition during the gastric digestion. The curd's collected at the end of digestion was also analyzed by the rheometer to obtain its rheological properties. And the figure shows the complex modulus of the curd. We found that from left to right, the complex modulus decreased significantly, which means the processing treatments changes the level of resistance to the curd deformation, and it shows in the following order. So we know the processing treatments can make the curd structure softer and easier deformation during the gastric digestion. Of course, the curd structure was also analyzed by the confocal microscopy. In the image, we found the red shows the fat, the green shows the protein, and in unhomogenized milk samples, we found the fat globules were evenly distributed in the protein aqueous phase. And in unhomogenized milk samples, the fat globule size is much bigger than homogenized milk samples. When the milk was digested for 20 minutes, we found a close-knit network of protein matrix was formed in all milk samples. Besides, the fat globules was also distributed in the protein matrix. With further digestion to 120 minutes, we found the fat globule size became much bigger compared to 20 minutes in all milk samples, which could be caused by two main reasons. One is the aggregation of fat globules. Another one is the coalescence of fat globules, because during the gastric digestion, the fat globule membrane protein could be hydrolyzed by the pepsin, and resulting in the coalescence of fat globules. At the end of digestion, we found the further coalescence of the fat globules could be found in all milk samples. And besides the curd structure in homogenized milk samples is much looser and with more pores compared to unhomogenized milk samples. The digester was also analyzed by the confocal microscopy. In undigested milk sample, they show the same picture as shown in the curves. And when the milk was digested within the 60 minutes, it shows significant difference in the structure. In unhomogenized milk samples, we found a large amount of protein, fat globules remained in the digester, whereas it's almost disappeared in the homogenized milk samples. On the other hand, in all milk samples, we found the protein almost disappeared in the digester, and except for some small protein particles in the homogenized milk samples. And at the end of digestion, we found some difference. And in unhomogenized milk samples, we found the amount of fat globules decreased slightly, and in homogenized milk samples, more protein particles could be found in the digester. So I made those conclusions. Compared to the digestion of cow milk reported before, we found the sheep milk could form the structural curves as found in cow milk during gastric digestion, and the processing affects the pH changes, the structures, composition, and the rheological properties of curd during the gastric digestion. And processing could also result in different composition and structure of gastric digester, and hence affect the delivery of nutrients to the small intestine. Here, I'd like to acknowledge the NZ3M project and its teammates and all the staffs and colleagues in Riddett Institute and Massey University. Of course, our industry partner, Springship Dairy and Maui Milk, certainly acknowledge the funding we received from MB. And thanks for listening.
Thank you very much for your interesting presentation, Zheng.
Yeah, thank you.
It's again, I thought it was really nice to see this really visual of the different curds and how different they are, just following these really simple processing things like pasteurization or homogenization that most of us just do without thinking too much about it. What Amber said earlier about how sort of the food structure affects the digestion, that's kind of the next step, and that's what the New Zealand Milks Mean More program is all about. So do you have any feeling for how, the softer or the harder curds, how is that firmness going to impact further digestion? Do you want to speculate anything about that, or is there anything known about if harder curds are digested more slowly? Or
based on our results so far, we can know the homogenization has a big impact on the sheep milk structure during the gastric digestion. It can make the curds more softer, as shown in my slides. The rheological properties of the curd decreased with the processing treatment, which means the curd structure has more large voids within the curd, which can be easier deformed under the gastric contraction. Yes.
Okay. Yeah. Okay, great. So I think the next few years at the Sheep Milk Conference, I think we're going to see quite a lot of presentations from the New Zealand Milks Mean More program.
Yeah.
So you might join us again next year. I'm going to move on to the final presentation in this session.
Thanks.
Thank you. So we've got Evelyne Maes, who is scientist at AgResearch based down in Lincoln. And as we heard in Amber's presentation earlier, she was talking about A1 and A2 cow milk. And I know a lot of us are kind of saying that, well, sheep milk is naturally A2 milk. And I think Evelyne is going to shed a bit more light here about the A2 sheep milk for us and make us understand this even better. So Evelyne, you can start.
Good afternoon, everyone. As Linda already mentioned, I'm part of the proteomics team at AgResearch, where we characterize proteins and peptides by means of mass spectrometry in a whole range of samples. This is from how I would say it, from farm to fork. And by characterization of proteins, that can be just checking some sequences, but it can also be protein profiles or modifications on proteins during processing. And today I will give an overview of a very small study that we did using sheep milk powder and characterizing beta-casein via mass spectrometry and see whether or not it's similar to the cow A2. So as Amber already quite nicely explained, proteins are key nutrients in milk, and beta-casein is one of the most abundant proteins in this milk in human and mammals. There has been some attention with A1 and A2 variants, and this is because the formation of BCM7, which is a peptide that is present in A1 variant of beta-casein, but not in the A2 variant. And this is because if we look at the amino acid sequence, then there is a proline that hinders the cleavage of the peptide. And this peptide, if it's released, again mentioned by Amber, can impact digestion and immune system. We know that sheep milk contains around 45% of beta-casein, which is quite a bit, and it has several genetic variants. So we were wondering what is going on here. And if we look at the beta-casein protein sequence just from the database, then when we compare to cow, we can see some similarities and some differences. So on top is the sequence of sheep beta-casein, the blue ones is a cow, and everything in yellow is the amino acid sequence, which is exactly similar. So we have a 92% homology, but there are some differences as well. And as you can see, so the first part of the sequence is also a Samuelsson peptide, which is cleaved off when the protein gets active. And then we have both in sheep and in cow, as is reported just sequence, the proline form, so the A2 form. And what we really want to know is whether or not this is actually true and what variants will do. And we use proteomics for this. So proteomics will look at the sequence and characterize this by first extracting the proteins. And this can be from milk, this can be from powder. So in this study, we just use powder. We extract the proteins and we really unfold them. Then we use an enzyme to generate tiny peptides. And when we have this whole mixture of peptides, we will separate them using chromatography based on their hydrophobicity. And this is according to time. So we actually, in the end, when it dilutes from the column, our sample is less complex. And then when it's diluted, we measure it with mass spectrometry. And just to give a very, very brief introduction into mass spectrometry, what we actually do is really showing that peptides that are eluted will be ionized, and then they will be separated according to mass. I can show it again. So the smaller ones will fly longer through the mass spec and will show pop up first in a mass spectrum. So based on these characteristics on mass and on charge, we can get some information on the protein itself. And our biggest question was, using this technology, we know that in cow milk there's an A1 and a 2 variant. And if we just look at the general sequence of sheep milk, it should be just the A2 variant. But is this true? And we tested it with 4 different sheep milk powders, 2 from New Zealand and 2 from Italy, and really just dig into the sequence. So no modifications, just in the sequence. And we used a few methods for it. So the first method is what we call shotgun proteomics. So actually, this is a snapshot of everything. So all the peptides that are present in the powder. And our peptide of interest was already a little bit interesting, because if we just digest it with our typical enzyme, which is trypsin, it gives peptides of a length of around 15 amino acids, apart from beta-casein, where it makes a peptide of 49 amino acids. This is quite long, and for a mass spec typically like shorter peptides to fly, is quite challenging as well. So there was a first hiccup in getting this peptide to fly. Well, we were able to let it fly. Then we were also looking at the other peptide variant, which has, instead of the proline, the histidine, and we were trying to find these in the whole mixture of all the samples. So what you can see here is on the top panel is a chromatogram of all the peptides that are eluting. And this is just, if you look back at the top panel, that's just a really simplified way of what these data actually are. So every peaks represents a few peptides that have similar characteristics and that are all present in the sheep milk powder. And because we know which peptide we were actually interested in, we can extract them from this really big pool of peptides. And because it's very big, we also know it has different charge states. So we can actually really extract the peptide of what we think is the peptide, so the mass to charge in different values. And if we do that, we can see that in our whole complex mixture, we're still able to really nicely see a peptide of our 3 mass to charge values that reflects the peptide that we are looking for, which is a very good start. But we need some more information. And what we did was then fragment our peptide of interest into smaller pieces just to really get this information on the sequence. So what you can see here is an MSMS spectra, as we call, with all these different peaks, which all reflect to these different fragments that are found here. And if we just analyze them all and sequence them all, then we have our large peptide of interest with some information on the sequence. And what we can see is that if we look at it, then we can definitely see that there's a proline there. If we look at the variant of which carried the histidine in the peptide, what we can see if we try to extract the values that are related to the peptide, we see some signal. It's already lower and it's really not nicely curved like we would expect from a peptide, which might indicate that it's not there. but we need a little bit more information just to be for sure that it's not there. So then we thought, oh, maybe this is just in one peptide, in one sample. But what we could see is that for the 4 different samples or 4 different milk powders, regardless of whether or not they came from Italy or New Zealand, we always get similar results where we have nice peptides extractions from the proline variant, while the histidine variant is not clear. Then in a second method, we really wanted to validate our data, because what we now have is a peptide that reflects the mass to charge, but we really want more proof, and we do that with targeted proteomics. So compared to the previous method where we really had a snapshot of everything, we now only analyze our peptide of interest. And what we also do is really looking at some of the specific fragment ions as well. And as indicated here, where we are really interested on is the ions that are related to this amino acid, which would give us indication on whether or not it's A1 or A2-like variants. So if we look at our targeted data, what we can see here is first a very nice peak reflecting our peptide. And then if we look at the masses related to the fragment ions that could confirm the sequence, we see that they're very nicely aligned with the peptide that we're looking for, hence giving validation that our fragment ions are there, so that the peptide is there, so that the proline is in the peptide, so it's an A2 variant. While if we look at the histidine variant, so the one variant, what we can see is that there is a small peak there, but very low intensity. So really saying there, if something of this mass would be there, it was not similar to a peptide that is there in A1. So if we just really quickly conclude these samples, then we can say, yes, with proteomics and mass spectrometry, we can characterize the sequence of the beta-casein. We had some hiccups with being at such a large peptide to ionize, but we were able to detect it. We did some targeted analysis really to confirm that this variant is present in all the samples of beta-casein. However, because there's only sequence homology and it's not completely similar, potential health benefits needs to be investigated independently. And with this, I know I'm quite quick, but it was a small study. So I think I would like to thank Lee and Stefan for their help, Ludovic Derry, of our stakeholder, and the proteins and metabolites team in Lincoln for the continuous support. And thank you all.
Thank you very much, Evelyne, for that nice presentation. Oh, I've got some sound feedback here. Sorry about that. Yeah, so what do you say? Do you say that we should not say that sheep milk is naturally A2? Should we say that sheep milk contains a protein that is that is similar to the cow A2 beta-casein, but it's not exactly the same?
Yes, or at least what I would say is we cannot say that it exactly has the same health benefits, because based on the amino acid sequence, we can see there are small changes between cow and sheep. So whether or not even the peptide does exactly the same between cow and sheep, that's something to confirm. But just based on that amino acid in that position, the histidine is not there. So in theory, it's an A2 variant. Yeah. Okay,
well, I think that your findings here will lead to some more interesting research if we want to investigate the further how this peptide that is produced from the sheep milk, and if that has the same effect as the cow A2 peptide that is produced. So yeah, that's interesting. Good.
Thank you.
Okay, so I have there are no questions coming in from the audience at this point in time, and also we have been running a little late here. So I think I'll say thank you to you, Evelyne, and I think all the presenters in this session have, it's been a really interesting session to try and understand this very complex food that milk is, and how it's digested, and how the smallest manipulation of the milk actually changes it quite a bit and makes a big difference to its nutritional, can make a big difference to its nutritional value. So I'd like to thank all presenters in this session for your contributions. Yes, I will hand over to Craig now to move into our fifth session for today. And I would also like to thank everyone who's online. We've got 150 people on YouTube, which is great. It's great to see the community coming together during these difficult times. So thank you all, and Craig, over to you.
Awesome, thanks, Linda. So I really appreciate your guys' session. I think that was a really complete story and a complete picture of where the science is up to, and I guess that the challenge now is to translate some of that into some of the products that businesses can think through, and what of those characteristics can be used to sort of contribute to the overall development of the industry. So that's really good.